So last lecture we talked about skeletal muscle mechanism, its contraction, excitation, contraction coupling. So today we'll
Yeah,
start with the characteristics of muscle contraction. So muscle contractor characteristics are divided into four different parts. One is the contractile and elastic component, properties of the contractile and elastic components of the muscles. Then concept about the muscle length, motor unit, and contractile response. So first, we'll talk about contractile and elastic components. Contractile component are component which presents moments of actin and myosin filaments or contractile proteins. जो भी मुझे प्रेजेंट का मैसेज करेगा मैं उसे एब्सेंट मार्क करूंगा. Uh, these protein are viscous nature, though we are saying thin filament, thick filament. Uh, it is not like that. They are viscous in the nature, and they doesn't have any elastic property. If you stretch, they will remain stretched. They will not recoil and come back to its original position. So it is like a more of like a plastic in nature. And whole muscle proteins out of this 60% is contributed by these contractile proteins. Then next component which is present in the muscle is series elastic component. These are the components, these are tissues which have elastic nature. So when they stretch, they can recoil and come back to its original position, and they have resistance to overstretching also. And these present not only in the entire muscles but also present in the tendon also. As we first class we saw the structure, the contractile portion, the myofibrils surrounded by the uh, endomysium, perimysium, all those things that together will form tendon also. So these are forms the component series elastic. Name series itself says they attach in connection with the contractile component. So there will be the contractile component, then series component. So they will attach uh, in total in a one line. And whenever there is a muscle contraction or relaxation or all occur, they affect the length of the series elastic component also. So like when there is isometric contraction occurs, what does it isometric? Iso means same, metric means length. So when the muscle is contracting but length is not changing, in that time it will cause a stretching of the series elastic component. Muscle is actually contracting, but it will cause stretching in the muscle contraction. That's why length is not changed. Though muscle contraction is occurring, length has been not changed, and it will only increase the tone in the muscle or tension in the muscle. So this is what actually series elastic component will be done. And once the contraction overs, it regains to its original position and also brings the contrary component also in its original position. The next component is a parallel elastic component. As name suggests, they lie parallel to the contractile protein, and they usually like connective tissues, sarcolemma, and other filaments are present along with the myofibrils. All those will represented by the parallel elastic component, and they are also in the elastic nature. So whenever there is a stretching occur, they will come back to its original position. Along with that, they also bring the contractile portion also to its original position. And this parallel elastic component usually shows more effect when there is a actual contractions occur. So that contraction is called isotonic contraction, where the tone of tension on the muscle will be remain the same, but muscle length will be changes. So that in parallel elastic component, that it will squeeze and the this component or fold up, and that shows the contraction. And this also offers resistance to the passive stretching. So when we stretch muscle passively, the resistance offered by this is both series elastic and parallel elastic. So together, series elastic and parallel elastic will almost contribute the 50% of total muscle protein. So when your muscle contract, Muscle contract in two different way. One of the mechanism of contraction is called sorry, isometric, means muscles length will be the same. And another type of contraction is isotonic, where the tone will be the same. Length will changes, but tone will be the same. So in these two, how we can explain the isometric and isotonic? Like when you are lifting your dumbbells or you are lifting anything, that lifting of the thing. Suppose I am lifting this phone, then this is the actually length of the muscle is changing, and that's why it come under isotonic contraction. Isometric when length is not changing. Suppose now you want to pull the wall or you are pushing the wall. Wall is a very uh, structure which cannot be moved very easily. So you are trying with your whole force, but the change doesn't occur. So muscle is not contracting. So length is not changing of the muscle. Length will remain same. The muscle contraction is going inside the muscle, but length will remain same. But the tone in your muscle will keep on increasing. That is the example of isotonic. So lifting anything is isometric, while pushing the wall is isotonic. Pushing something नहीं, जिसको push कर पा रहे हो, then that will also isometric. Suppose something you want to lift and you cannot lift, then that contraction is also isometric. And if you able to lift, then it is a isotonic. So this is how what is the difference between isotonic and isometric. Now what will happen? This is if you see this three diagram. Okay, if you see these three diagrams, then this is the first example of when muscle is at a rest, normal length. The second diagram is showing when there is isometric contraction. So length of the muscle is not changed. So length of the muscle will be remain same, but the actual contractile protein contracted. So the if you see this length, contractile protein is contracting and it is creating the contraction within the muscle. But actual muscle length is not increased. So that water length changes, it compromised by the series elastic component. It will cause a stretching on the series elastic component, and that will increase the tension in the muscle, and that will brings to increase in the tone of the muscle. At the same time, it will also cause a folding of the parallel elastic component. Now come to the isotonic contraction. This is where actual length changes when muscle is contracting. In that time, the series elastic component will not stretch. It will have its original length and it will move along with the muscle contraction, and the parallel elastic component will fold up and it will actually reduce the length of the muscle. That is how the isotonic contraction occur. And the C is the example of passive stretching, when we stretch muscle passively. How can I explain the example of passive stretching? Exercise के बाद आप stretching करते हो, you do like something like this or something like these muscles. So this passive stretching will stretch your muscles, and that will cause a stretching on the both parallel elastic and series elastic component, and they will bring back the muscle to its original position. अगर एक series elastic और parallel elastic component नहीं होते, तो once you stretch muscles, muscle will be completely lengthened. It will not come back to its original position. That doesn't happen because of presence of both series elastic and parallel elastic component. These both component also provide resistance to the muscle contraction. जब भी आपके muscle contraction होता है, तो it provides little bit inertia and resistance to those muscle contractions. So contractile protein has to overcome the resistance offered by these elastic components also. Now come to the concepts about the muscle length. There are four different terms we use. One is optimum length, second is resting length, third is equilibrium length, and fourth is initial length. Optimum length is the length of the muscle which can provide maximum contraction or maximum tension. अगर उस लेंथ से मसल कॉन्ट्रैक्ट हो जाती है, तो that will provide maximum length. And this optimum length is the length when the distance between myosin head and myosin binding site present on the actin filament is very small. So when we slightly stretch it and we'll make the sarcomere length nearly 2.5 micron is the optimum length of the muscle. But resting length is not same as that of the optimal length. Resting length is a length of the muscle when muscle is not stimulated at all, and it is at the natural body position. Objective natural body position means now this is not a resting length, or this is not. When you sleep, where I will lie down and you put your both arm on your thighs, or you're close to the thighs, and the whatever the that time of the length of your limbs and upper limb, lower limb, those length of the muscle are called resting length of the muscle. And normally muscle contract from this resting length only. And for many muscles, the resting length is almost equal to the optimum length of the muscle. The third concept is come about the equilibrium length. Now, when we are growing, during that time, the muscle is also grows and bone also grows, but bone grows little bit more as compared to muscle. So during even at the rest, muscle is little bit at the uh, stretched. So, but if you cut the both end of the muscles from the both insertion and you allow, it will contract little bit. That is, even if muscle is relaxed, then it will reduce its length. So, the actual length of the muscle when there is a no uh, ends will not be tied to anything. They are free ends. That length is called equilibrium length, which is always smaller than the optimum length and resting length. Then come to the initial length of the muscle. It is any length of the muscle uh, which is just before the contraction of the muscle. It's called initial length. Could be length. It can be optimum length, it can be resting length, or it can be equilibrium length. It can be overstretched length, or it can be squeezed muscle length. Whatever length of the muscle, just before contraction, whatever the length of the muscle is there, that is called initial length of the muscles. Now, remember this initial length of the muscle is very important term for when we are talking about the effect of the load on the muscle. Okay. Then come to the motor units. So motor unit is the number of the muscle fibers supplied by the single neuron cell body. So, depend upon the different body part, like muscles of the trunk, lower limbs, they will have more muscle fibers with a single motor unit as compared to muscles of the fingers or small muscles, but a very less number of the muscle fibers is supplied by the single nerve body. These whole together will form motor unit. There are two different type of the motor units: slow motor unit or fast motor unit, also called as a red motor unit or white motor unit. Red, they have little bit reddish color, and it is because of presence of myoglobin. So myoglobin provide oxygen and all those things, and that provide immediate oxygen to the motor unit, and they can work for longer time, even if when there is a less oxygen. These are called uh, slow motor, red motor unit, and they usually utilize more of the anaerobic metabolism as compared to white motor unit. While white motor unit has a little bit low concentration of the uh, myoglobins, and that's why they shows a little pale color, and they are very much fast, and they usually utilize more of the anaerobic type of the metabolism as compared to red motor unit. So uh, depend upon the number of the motor units, sometimes we can divide muscle fibers also into fast and slow muscles proper, like muscles of the eye, which are very fast to act. So they're usually made up of the white fibers, while muscles of the trunk or back, which are slow to act, they're usually made up of uh, red motor unit. And with also exercise and type of the pattern, also we can increase and decrease component. The many muscles are mixed, has contain both red and white motor units also, and we can increase or decrease the concentration of red or white motor unit. So this is a difference between the red and white motor unit. Can go through it. Um, okay, then come to the contractile response. So how the contraction occurs? I said there are two types of the contraction: isometric and isotonic contraction. Then in experimental animal, we can do single muscle pitch. Then we can see the various factors which affect the force of contraction. So isometric contraction is a when the length is remain same even if the muscle is contracting, but tension will keep on increasing. That type of the contraction is called isometric contraction. Since there is no object, no movement is occurring, So there is no external work done. This causes shortening of the series elastic component, but it causes shortening of the contractile component, but it will cause the stretching of the series elastic component. Example, like I was already pushing the wall or maintenance of the body posture against the gravity. These are example of the isometric contraction. Then come to the isotonic contraction. Here the tension will remain the same, but the length will keep on increasing. As the length is changing, it will bring the external work done. Here both contractile component and paraelastic component will is shortened, while stress elastic component will remain same. Example, lifting of any object, walking, running, or flexing of your arms and all those things that will come under the, or whatever, any movement which, any muscle contraction which can cause movement is a example of isotonic contraction. So now these both isotonic, isometric difference we could understand, but many times this is not in isolated form, that there is only isotonic type of contraction or isometric type of contraction. Suppose this is a water bottle. I want to lift it. Now this water bottle is present on the table and I am lifting it. Okay, this is pure example of isotonic contraction. Now instead of water bottle, water, it filled with the mercury. इतने size का होता है, but it is a very very heavy. Now I don't know mercury is heavy, and I try to lift it. So when I lift, I feel that I could not able to lift it, and there is a I need more force. During this time, muscle is not actually shortening its length, but muscle is contracting. So tone is keep on increasing. So initially it is a isometric contraction. One point will come when we judge the uh, brain will judge the actual weight and how much force has been applied. Slowly it will keep on activating more and more motor in it. And one point will come when enough force has been developed, then muscle will contracting. So first initial portion is a isometric, and then it will become isotonic. So there are many times a mixture of the moment of contraction can also occur. So it is not like in isolated form that there is always either isometric or isotonic. It can be both isotonic and isometric. Like weightlifting also, when you are doing weightlifting, that time also first it is a isometric, and then that weightlifter will able to lift it, that it will become isotonic muscle contraction. Then come to single muscle twitch. This doesn't happen in our normal body. Now makes clear this, because many times we'll consider, because this is the graph which we taught so times, we consider this is a normally present in your body. This is just experimental, and the simple muscle twitch only occur in experimental animal. When it normally occurs, it will cause a fluctuation. So, कभी आपको लगता है यहाँ पे कुछ हो रहा है, यहाँ पे कुछ हो रहा है, or sometimes these small muscles there is a contraction is occurring. That fluctuation occur. That is an example of simple muscle twitch in normal body. Otherwise, normally simple muscle twitch doesn't occur. Okay, it occur in we do this simple muscle twitch in experimental animal. We'll take frogs, dissect the frogs and remove the gastrocnemius muscles along with the sciatic nerve, that is called gastrocnemius sciatic preparations, and we'll put that uh, uh, gastrocnemius sciatic muscle. We remove the gastrocnemius muscle along with the bone. तो femur bone उसका साथ में हम लेते हैं, and so there is a instrument on which the will mount this muscle. So bone will put in that hole, and the muscle will tie to this, and this another end of the muscle will tie to the pointer, and that pointer is attached to the graph. So whenever the muscle contract, it will causes movement of the pointer, either upward movement or lower movement, depend upon here pulley will be there. So it will pull downward, it will cause upward movement, and this is how it will show muscle contraction. So this is how the assembly will be there, and here the nerve we supplied here will put on the electrode. So electrode पे हम वो nerve रखते हैं, and then we'll stimulate the nerve. Now once you stimulate the nerve, so site where it is stimulated, or time at which it is stimulated, that is called point of stimulus. So it is a time at what time it is stimulated, and then it after a fraction of second it will initiate the contraction in the muscle. So time taken from the point of the stimulus till the initiation of the contraction of the movement is called latent period. So it is a start of contraction phase. Okay, this whole period is called latent period, and this latent period is a time taken for the various things which happens. First thing, it is transmission of the impulse from the nerve to the site of the neuromuscular junction, then release of the neurotransmitter in the neuromuscular junction, then generation of the action potential in the skeletal muscle at the neuromuscular junction. Once action potential generated, it release of the calcium, that excitation contraction coupling to occur. Once the excitation contraction coupling occurs, sliding phenomena will occur, and it also to overcome the resistance of the series elastic component, parallel elastic component, and that will develop the tension in the muscle, and then lifting of the liver. Liver also has its own inertia, so depending upon the liver lifted, so all those time will take, and then liver lifted, that will initiate the phase of contraction. All those time will take period, and that is called latent period. Once the muscle contract, it will goes to its maximum contraction. So this is a phase of maximum contraction. Then it start relaxing. So this is a relaxing phase, and once it comes to the maximum relaxation or maximum relaxation. Once it reaches to maximum relaxation, again it goes up. This curve is called shattered curve. It is because of that liver fall under the pulley and it bounce back. Because of that, another upward movement will be there, and this shows that this is a experimental contraction. Okay, and this is how the uh, simple muscle twitch we can describe. Here, the duration of the simple muscle twitch varies with the different fiber. Fast fiber will be very small, that is about less than 0.02 second. Where slow fibers will have a little bit longer contraction, that is 0.1 second. Length of the contraction and relaxation, all those things will depend upon type of the muscles, and depend upon the stimulus also. Stronger stimulus can produce strong contraction, smaller stimulus can produce less contraction. Okay, then come to the various factors which affect the stimulus, affect the phase of contraction. Lots of experiments are done. It is not possible for me to describe each and every, but I will try to describe various experiments. So first factor is the strength of the stimulus. So what we'll do, we'll keep on increasing the strength of stimulus and we'll start stimulating the muscles. So how we can increase the strength of the stimulus? जिसने मेरा एक्शन पोटेंशियल का पहला लेक्चर देखा होगा उसमें पता होगा. We have used the stimulus induced current. So this is primary coil which attached to the battery, and this is the coil, and there is a secondary coil which attached to the electrode. So this is the electrode. So whenever we'll switch on the circuit, it will initiate the electric current to the primary coil. That will generate the magnetic field. That magnetic field will generate the electric current in the secondary coil, and that will stimulate the muscles. Now here induced current will be very momentary, because it will develop the current only with the change in the magnetic field. Once the magnetic field become constant, there will be no current flow. So जब switch on करते हैं, there is increase in magnetic field that will generate one current, and that will stimulate the muscle contraction. Then we break the circuit, that will reduce the magnetic field, that again produce a current. Here when we are making it, the change in the magnetic field will be a little bit slower, so current induced in the secondary coil will be smaller. So make current is usually smaller current. When you break the circuit, that change in the magnetic field is much faster, and that will induce little bit higher current. So break stimulus is having little bit higher stimulus. So understand this, make will be as a low current, while break stimulus will have a high current. So that high induced current, that will keep on doing this, and the strength of the current also depend upon the distance between these two coils. So what we will do, will put them apart and will change the make and break, and will keep on stimulating. One point will come which initiate the muscle contraction. That is a threshold stimulus. And that initiation will occur only during the break situation. Make will not produce any muscle contraction. Okay. Then we start decreasing the distance between the two trial and keep on stimulating. So we're slowly increasing the strength of the stimulus, and now even make stimulus can also initiate the muscle contraction. But the force of contraction will be keep on increasing as the distance between the two coil keep on decreasing, as the strength of the stimulus will keep on increasing. One point will come when almost all motor unit gets stimulated. Now there is no more motor unit available. So all motor units get stimulated, and it will causes complete muscle contraction. That is a phase of maximum muscle contraction. अब उसके बाद आप कितना भी स्ट्रेंथ बढ़ा दोगे उससे ज्यादा मसल कॉन्ट्रैक्शन नहीं होता है. सो हाउ दिस मसल कॉन्ट्रैक्टिंग, पहले बहुत कम कॉन्ट्रैक्ट हो रहा था, फिर और ज्यादा और ज्यादा और ज्यादा बढ़ते जा रहा था. तो हाउ दिस मसल कॉन्ट्रैक्शन, फोर्स ऑफ मसल कॉन्ट्रैक्शन कीप ऑन इंक्रीजिंग विद इंक्रीजिंग द स्ट्रेंथ. जब हम कम करंट स्ट्रेंथ स्टिमुलेट करें तो इट विल जस्ट स्टिमुलेट द फ्यू मोटर यूनिट्स जिनका थ्रेशोल्ड बहुत कम है, वो motor units activate होते हैं. There are only few motor units contracting, so only few muscle fibers has been contracting. That's why they generate very less force. As we keep on increasing the more and more current, more and more motor units will get stimulated, and that will start initiating more and more force of contraction. This mechanism is called recruitment of motor unit. Okay, and same effect we'll see in normal muscle contraction. As I said, this is a water bottle. I'm lifting it. Okay, now this is a mercury bottle. When I try to lift, I feel it is very light. Then hardly few motor units will be activated. And now when I want to lift it, now I know the weight is more. More and more motor units start activated. So we are recruiting more and more motor units to generate more and more force contraction. When the force of contraction become little bit more than the actual weight of the object which I am going to lift, then only muscle will change its length. Okay, this is how the recruitment phenomena work. When you are lifting a smaller object, the less motor unit will activate. When you are lifting heavier object, then more motor units will be recruited. Okay, so this is how the strength of the stimulus and uh, motor units will work to increase the strength of muscle contraction. Then second phenomena is the effect of frequency on the stimulus. Now, what we'll do, we'll keep on stimulating the muscles at a constant rate of the contraction. Before understanding this, we should understand the effect of two consecutive stimulus. Now, suppose this is a simple muscle twitch. This is a simple muscle twitch. And now what I will do, I will apply the second stimulus here. Okay. Here, this is a PS1. Here, PS2. Second stimulus will apply. And that will again show the contraction of the muscle. But here the second time when water contraction occurring with the same strength of the stimulus. Now here we are not increasing the strength of stimulus. That will shows little higher concentrated contraction as compared to previous contraction. It is because of the effect of already previous contraction. So पहले जो contraction हो उसकी effect की वजह से second contraction is little bit higher, and that is called beneficial effect of first stimulus or beneficial effect. And this beneficial effect include there is already calcium is already present in the muscle. All calcium has not been removed, so already there is a calcium ion present in the muscle. Our next stimulus will provide more calcium to that muscle, so it will causes more force of contraction. Second thing, already previous metabolism increase the temperature of the muscle, and that increase in temperature of muscle will reduce the viscosity of the sarcoplasm, and that will reduce the resistance or inertia of the muscle. That will be reduced. So resistance offered by the series elastic and parallel elastic component will also be reduced because of decreased viscosity of the sarcoplasm. So that is another point which help in a better contraction. Third, reduction of the inertia of the liver. Liver is already in the moment, so that inertia has been reduced, and then that why also liver is lifted very fastly. So these are the few example of beneficial effect occurring with subsequent stimulus.
Now what we'll do with next contraction, what we'll do, instead of putting the second stimulus after relaxation, we'll put second stimulus somewhere in a relaxing phase. What will happen, then muscle will contract here, and from here it again start contracting. Sorry, again it start contracting here. So height of contraction is higher because of previous beneficial effect, but muscle is not relaxed uh, completely. Then what we'll do, we'll give stimulus to the phase of maximum contraction. So हमने frequency कम कर ली. पहले suppose we're talking about, suppose we'll talk about the slow muscle fibers. There first we give after 0.1 second. Now here we are giving after 0.075 second. Now here we are giving after 0.05 second. So 0.05 second पे हमने stimulus किया, then there is a contraction. So this muscle is not relaxed and it is contracting. Okay, that will be the effect of when we are reducing the length. Now we again reduce the or increase the frequency and reduce the distance between two stimulus, and instead of 0.1 second, here we are giving 0.025 second per stimulus. So it comes somewhere here, and when we are stimulating, it is just during the phase of the contraction, that will again causes more contraction. So this is how it will. Now it will be only one curve. We will not see two curve, but that second curve is much higher than if we are giving in the first stimulus. And now if we are giving in the just suppose 0.001 second, now here this muscle is refractory and muscle is not responding. Now the muscle is contracting with the same phase of the contraction. So this phase refractory phase is also there, but the refractory phase is very small. And this is how the two consecutive stimulus when we apply, that will show little bit effect on the second contraction of the muscle because effect of previous contraction. Now same effect we can see if we keep on stimulating repeatedly. Suppose we are stimulating, we are talking only about slow fibers, and we are stimulating with a continuous stimulus with a frequency of gap between the two stimulus is about 0.1 second. Then muscle will contract like this. Okay, jerky movement will be continuously occurring. Then if we stimulate in somewhere around 0.07 uh, 0.075, that time there will be the jerky movement will be there, but muscle will not relax completely. But if second stimulus will occur just in the phase of contraction or just at the end of the phase of contraction, so muscle is just completely contracted and then second stimulus. So if stimulation is given by 0.05 second, then muscle will not relax at all. It will always in the phase of contraction. So relax होने का time नहीं मिलेगा और phase it keep on contraction. And that phase will called a titanization. So it will development of the tetany. So जो first यहाँ पे दे रहे हैं, then there is a trap. We call it a trappy phenomena. So muscle will have little bit higher and higher contraction, but it is also relaxing also, though it doesn't completely relax. When we are giving in between the contraction relaxation phase, we call it incomplete relaxation. There is no complete relaxation. Always muscle is in a phase of partial contraction, but movements are jerky. But once we give it in the phase of contraction, then muscle will not relax at all, but it will always in the phase of the contraction. And that mechanism is called tetanus. The frequency at which we can initiate the tetanus is called a critical frequency of the tetany. For the particular muscle, there are different tetany. Like if we are giving for the small, fast muscle fibers, then we might have to decrease it up to 0.00125 or something like that. And that will be the critical frequency at which the tetanus can develop in that particular muscle. And that will lead to contraction of the smooth, sustained contraction of the muscle. And this is how the tetanization we can develop into the muscle. And now when we see the word tetanization, we always feel it is like a pathology. Tetany हमने disease पढ़ लेते हैं. Tetanus toxins we are listening. That Clostridium tetani we are listening. So we always refer it as a pathological condition. But tetanization is a pure physiological condition. जो आपके बॉडी में जो भी quality muscles contractions हो रहे हैं, all those contractions occur because of tetanization. Now when I'm lifting this, then it causes smooth sustained contraction of these my shoulder muscles and
elbow muscles, and that is because of continuous stimulation going to the shoulder muscles and elbow muscle that will allow me to keep this contraction sustained at particular position, and that is also smooth and sustained contraction. So how we can define the tetanization or complete tetanization? Smooth and sustained contraction of the muscles when stimulated by the frequency of the stimulus which is more than critical frequency. Okay, this is how the titration, and if we keep on increasing the critical frequency of the stimulus, more and more motor unit will be recruited and more strength can be developed by those particular muscles. Okay, then come to the effect of load on the muscles. So it is, if we apply the load before contraction, then we call it as a free load, and if we apply the load after contraction, then we call it as a afterload. So okay, nahi, so now again we'll make that so this is how that now muscle preparation will be there. Muscle लगाई है यहाँ पे and attached to the liver. So liver is also put on the nail here. Liver is pulling on there. and यहाँ पे हमने load लगा दिया, weight लगाया है यहाँ पे. Okay. So if this load act on the muscle before contraction, then we call it as a free load. अभी यहाँ पे हमने nail लगा रखा है. Okay, sorry, यहाँ पे हमने इसको support लगा के रखा है nail, and that's why it's resting on the nail. So load is not acting on the muscle. And as the muscle contract, it will pull this down and that will lift the load. Now muscle is not resting on the nail, and now load start acting. So this is how it is after load. If we remove this nail, and now the weight is actually acting on the lever and it will pull little bit downward. So it will movement will be something like this. Okay, and the load is acting before giving stimulus. So muscle is not contracting before contraction, only muscle is acting. That is called free load. Okay, understand between free load and after load. Now what happen in free load, it will cause little bit stretching on the muscles, and that will bring that muscle in optimum length. And if we stimulus on this, the muscle will contract with its full force of the contraction. So force of contraction generated in free load is much higher as compared to after load. And this phenomena is referred as a Starling's law, Frank-Starling's law. So what Starling suggests, within physiological limit, the force of contraction of the muscle is directly proportional to its initial length. So force muscle contract करने से पहले हमने लोड लगा दिया तो लेंथ बढ़ गई मसल्स की, that will generate more force of contraction. Similarly, after load say there is no change in the initial length of the muscle, and that will keep on decreasing the force of contraction because load is acting after muscle is contracting. Now, how we can see this in day-to-day routine life? What is a free load and after load? So this bottle is lifted, kept here on the table, and when I hold it, and when I lift it, so the weight is occurring on muscles after muscle is contracting. So this is example of after load. But suppose I'm carrying this bottle in my hand, and now I want to lift it. So यहाँ से मैंने यहाँ तक lift किया, तो this bottle is already acting on a muscle before contraction, and that is example of a free load. So जब हम bottle carry कर रहे हाथ में, we're walking on the road, that it is muscle is not contracting. Now I want to lift it, so muscle is contracting. So load is acting before contraction. That is example of. So carrying water bucket is a example of free load, while lifting the water bucket is example of afterload. Okay, so this is the effect of load. This is how the Starling's law we can we can describe. Then effect of temperature. At low temperature, it will decrease all metabolic activities in the muscle. And that will delay the latent, increase the latent period, delay the phase of contraction, phase of relaxation, and also reduces the height of the contraction. While increasing the high temperature will fasten the process, will shorten the latent period, shorten the phase of contraction, shorten the phase of the relaxation, also increase the height of the contraction because of decreased viscosity of the muscle. But that is also within physiological limit.
After some time, mm, if temperature goes beyond this limit, that will start denaturing the proteins of the muscles and can prevent the contraction of the muscle. So when you are talking about low temperature, it doesn't mean that 0 degree. It is just 7 degree plus minus. 37 से 7 डिग्री बढ़ा लिया, 44 डिग्री is a high temperature. So 37 degree से 7 degree कम कर लिया, 30 degree is a low temperature. So we are talking about only within this phase how it affects the muscle contraction. So that's all properties of the muscles and characteristics of the muscle contraction. Then come to the muscle tone. Physiologically, we can define muscle tone as a state of the partial contraction of the muscle at rest. Okay, we'll take a 5 minutes break and then we start this. Okay, now come to the muscle tone. Muscle tone is a state of the partial contraction of the muscle when muscle is at rest. So आपके अगर जो muscle body में even if you are at completely rest, muscle is not completely relaxed. It is always in a state of partial contraction. This is also called a partial tetanus, state of partial tetanus, and it is because of the continuous discharge coming from the gamma motor neuron. This gamma motor neuron is the neuron which supply to the muscle spindles, and that will keep on activating the muscle spindles, muscles contracting, and that will initiate the stretch reflex and activate the uh, alpha motor neuron and will cause the muscle little bit in the phase of the contraction, and that will increase the muscle tone. So this is how the muscle tone is maintained in our body. If there is no nerve supply, there is no gamma motor discharge, and muscle which doesn't have any nerve supply will not have any tone. So it is not like a brain is keep on sending impulses. It is it's on its own impulse, and the muscle which contain more muscle spindles will have more tone as compared to muscle which contain less muscle spindle. So muscle spindles are more, you will see in anti-gravity muscles, like muscles of the flexors of the extensors of the knee joint, hip joint, muscles of the back. These are anti-gravity muscles. They contain high muscle spindles, so tone in those muscles are much higher as compared to rest of the muscles. Then graded force generation in the muscles, how the force, uh, same muscle can generate small force and also high force, and it is depend upon the three different factors. One is the recruitment of the motor units, how many motor units are involved, that will decide the force. Then frequency of nerve impulses, the titanization, more is the impulses generated, that will decide, and also synchronization of the impulse. So muscles having similar actions, if more muscle has been stimulated, then it will cause more muscle contraction. So these are three factors which are important and can generate the force of muscle contraction. So recruitment, there are different motor unit has a different threshold stimulus. So जब बहुत mild stimulus intensity देते हैं, then only few muscles will be contracted, and that will generate the force of contraction. When more and more strength of the stimulus will apply, more and more motor end will be keep on recruiting, and that will produce more and more of force of contraction. Second thing, at what frequency that impulses is coming, and that will generate the titanization in the muscles, and that will produce the force of contraction. So higher the frequency, more titanization and more force of contraction will be generated, and that will also recruit more motor unit also. So high frequency impulse will recruit more and more motor unit, and will generate titanization in more motor units, and that will produce more force of contraction. Synchronization of muscles, so impulses all, so different motor units will be there. If they stimulated simultaneously, then more and more force of contraction will be generated. If they develop all simultaneously, then less force of contraction will be generated.
Then come to the muscle fatigue. Fatigue जो हमारे दिमाग में होता है, that is different. Muscle fatigue is different. It is temporary loss of ability of the muscle to contract even after you apply the stimulus. This is usually occur after repeated stimulus. So if you keep on stimulating the muscles, it will keep on contracting, 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 contracting. One point will come when it will reduce its response to the stimulus, and slowly it will completely stop responding to the stimulus, and then we call it as a muscle is uh, able to muscle is fatigue, and this is called fatigue. They will not able to maintain the tension of the muscle contractility. This is called fatigue. This condition is temporary, so once you give rest to the muscles, it will regain its ability to contract. So fatigue is temporary. It is not like a paralysis where you complete loss of muscle contraction. Okay. Now, how that fatigue develop in the muscle? So there are different sites where fatigue can be developed in uh, entire living human body. The site of the fatigue where actually fatigue occur, it is in the central nervous system. So synapses where the neurotransmitter is exhausted, and that can generate the fatigue. And this fatigue can be overcome by just simple motivation. Even if you motivate, then that fatigue can be overcome. So it has a psychological factor also. Suppose you're doing some work which you enjoy, then it can delay the fatigue of the muscle. Uh, Central nervous system fatigue. If if any work you are doing is not interesting, or if you don't like to that work, then it increases the chance of fatigue. Then early fatigue will be set in that work. So it also has a psychological effect. Similarly, motivation. If someone motivate you or encourage you to do the particular work, then you can delay the fatigue also. So this is also what where central nervous system is involved. Second side of the fatigue is a fatigue will occur at the neuromuscular junction. So here, when we keep on stimulating the neuromuscular junction, one point will come the neurotransmitter present in the synapses that will be exhausted. Now there is no more acetylcholine available in the neuromuscular junction, and now muscle will stop responding to the stimulus. So second side is this neuromuscular junction. This you can see even in human being also, and also in experiments, live human being also, and in experiment nerve muscle preparation also. We'll see this is the site where actually fatigue occur in nerve muscle preparation. Third site when fatigue is actually occur in the muscles. After repeated stimulus or repeated contraction, it increases the metabolism in the muscle, and anaerobic metabolism will also increase and start accumulating pyruvic acid and lactic acid in the muscles, and that can cause a fatigue into the muscle. Decreased calcium content in the muscle, that can also causes a fatigue in the muscle, and depletion of the glycogen in the muscle, that can also decrease in the muscle. But our body will not wait till that the muscle will get fatigue and there is increased acidity that can damage to the muscle. We start getting fatigue at this level only, central nervous system. Even it is doesn't occur in the neuromuscular junction also. Okay, so this is how the fatigue will be. We can grade, and it does have psychological component. And how we can test for the fatigue? One is the ergography, is the experiment where we can do fatigue in a live human being, where the weight is attached to the finger. We'll ask him to keep on contracting the finger till he can do. Then we'll ask him to motivate him, and we'll see that even after motivation the work done will be increased. And then another experiment we'll do here, will ask him to perform the work done with a regular rest. So we'll ask him to do it for 30 seconds, then take a rest for 10 seconds, then again 30 seconds, then take the rest for 10 seconds. Then we'll find that fatigue we can delay. Work performance will increase when we are doing rest and work, and rest and work. And same thing you can do for your study also. You don't study at a stage for 5 hours. Take a study for 30 40 minutes, then take a rest for 10 minutes, then again study for 30 40 minutes, then take rest for 10 minutes. That will increase your duration for the study and your performance and concentration ability also. Then come to the disorders of the skeletal muscles. One is muscular dystrophy, where it leads to slowly increase in uh, weakness of the muscle, and it is because of the defect in proteins. structural proteins which present in the muscle, uh, and it is genetic disorders. Then second condition is myotonia, where the sodium chloride channel has been defected, and that will abnormal relaxation of the muscles or prolonged relaxation of muscle when muscle is voluntarily contracted because of abnormal sodium channels, and that will increase sodium entry into the muscles. Then abnormal tone, it can either increase or decrease in the muscle tone. When we increase in tone, we call it as spasticity or rigidity. When we decrease in tone, we call it as a hypotonia. Then come to the another condition is called degeneration hypersensitivity. This occur when muscle is paralyzed or muscle lack the nerve supply, and that muscle is paralyzed. Because of the paralysis, there is no more neurotransmitter release in the motor end plate, and that will causes upregulation of the receptors for the neurotransmitters. And now there are too much receptors present in the motor end plate. And now when renervation occur, or if somehow the acetylcholine will release, these muscles are very much sensitive for the even small dose of the acetylcholine also, and that will cause a severe contraction or spasmodic contraction, or irregular contraction, muscle twitch can occur in those cases. So irregular contraction can occur, and this is called denervation hypersensitivity. The acetylcholine might become from any blood supply or any other thing that can cause muscle to irregular contraction of the muscles. Then energetics of the muscle contraction. The first source of the energy for the muscle contraction is a hydrolysis of the ATP. Then ATP, second source is a resynthesis of the ATP by creating from the creatinine phosphate. The creatinine phosphate will release energy, and that energy utilized for the formation of the ATP. The second source of the formation of resynthesis of ATP is through the glycolysis, where the anaerobic metabolism will occur and it convert into 2 ATP and 2 pyruvic acid will be there. And in the uh, presence of the oxygen, aerobic metabolism occur, that pyruvic acid goes into the Krebs cycles and can provide the energy. And third form is oxidative metabolism, that is uh, from the Krebs cycle, and that's a major source of the energy. And we can keep on working the muscles if we utilize only aerobic metabolism. And that's why we usually ask people to do aerobic exercise for reduction in the weight, so that they can spend energy uh, for longer time. So there one concept is about oxygen depth. It is like when you are doing intense exercise, a lot of oxygen is consumed, but also body utilizes the energy without oxygen, and that will increase demand of the oxygen even after we stop the exercise. This is called oxygen depth. So this is how the oxygen deficit which create when we are performing the exercise, and that oxygen deficit is created by the uh, utilization of the creatinine phosphate, utilization of the oxygen from the myoglobulin, and doing anaerobic metabolism that will create the oxygen deficit. And once we stop in exercise, we keep on hyperventilating, and whatever the oxygen were taken from the for creatinine phosphate metabolism will recreate the creatinine phosphate, will provide oxygen to the myoglobulin which present in the muscles, and also whatever anaerobic metabolism is done, lactic acid and pyruvic acid will be, that will again goes back to the aerobic metabolism. Krebs cycle will be there, and that will replenish what oxygen depth has been developed. And this is how the oxygen consumption and that oxygen depth will be there. Here, the demand is more as compared to oxygen supply. So oxygen consumption is much more as compared to oxygen supply, and this is how extra oxygen will be supplied after exercise. So that's why you even start hyperventilating even after stoppage of the exercise. But here, understand the hypoxia never created. Hypoxia might be created at the level of the muscular level, but in blood, hypoxia never created. So this is how this oxygen depth will be we can justify. Then effect on the pH will start decreasing. Normally it is alkaline pH when at rest, but as the muscle start contracting, it start becoming going towards the acidic pH, and that will recover when at rest. Again it recover and bring back to normal pH.
Then in heat production, at rest muscle will keep on doing the basic heat production. But once we start contract the muscles, it will start producing the heat production, and isotonic muscle will produce more heat, while isometric produce as low heat will be there. And that's why the shivering and all those things will produce much more heat, and that body will start recovering after muscle contraction post exercise. Will take a rest, and that heat will be dissipated and goes to the rest of the body and release in the form of the sweating. Even if when we relax the muscle, that time also we start producing the heat also. So contraction also produce it, and relaxation also produce the heat. That's all about the muscle contraction. Any questions? Any doubt?